Great. Thanks so much, Inez, and thanks, Marina and the Stellenbosch team for hosting us for this week here. It's been really amazing. And then, as Inez mentioned, my group really at the interface of physics, engineering, and maybe material sciences. And so it fits nicely from the previous talks in that we're looking at the roles of not pure chemicals, but rather perhaps the physics or the physical chemistry of interfaces. So what you see here is some kind of artist's impression of a spiky surface on perhaps a bio-inspired material. And so here we'll try to cover the perhaps macroscopic aspects of wetting and spreading and maybe even physical interaction between topography and the biological membrane. So that's us at Imperial. So we're a university in the middle of London, and this is next to a bunch of museums. And the reason I share this is that here we have the Natural History Museum, and here we have the Science Museum. There are a bunch of other museums, but they have an incredible collection of specimens of plants and insects, and these date from some of those from several hundred years ago, and they have spares, so they often give us bits of wings and cicadas and carcasses of things, and so we can look at them, put them under SEMs in destructive techniques, sometimes focused ion beam techniques, etc. And so we are in this kind of a special place in that sense. So this is it. So these are examples of topographies of wings of, say, cicadas, dragonflies, etc. And the reason why they're relevant is because in nature there are plenty of examples of highly antimicrobial surfaces that just don't seem to use any antibiotics. They don't seem to have any trouble getting rid of bacteria, microbes, etc. from the surfaces, and they do that at scale, right? So clearly, if they can do it, we can not perhaps do it as well, but we can try to copy them. So you can think of this as in many ways, in many points of intervention. You could think, well, before a biofilm forms, the first thing that needs to happen is that, well, they'll need to come in contact to the surface in the first place. They'll need to attach. And then you'll know this better than I will. Eventually, there are a bunch of steps that will lead to that kind of, I guess, the nasty slime in the tap that I think you were describing earlier. So the question here, without going through the details, is to see, well, can we use topography to intervene at many of these stages? So let's go sort of one at a time. Maybe can we stop that initial attachment by, for example, making, let's say, a respiratory droplet not even want to stay on the surface, just simply bounce right off. And so you don't need to worry about the subsequent stages because, well, it just won't be there. For example, in the case of as mentioned in the bus demonstration, we are applying these onto the windows, for example, such that if you were sneezing and you turn over onto the wall, then you will not get that kind of deposition straight away. And then you can get the idea, right? Maybe there are other topographies that can act initial stage, but at subsequent stages. Nature, of course, does that. So in the first stage, you can see that there are surfaces here, for example, a lotus leaf that has this incredible superhydrophobic properties. They are known to come from their topography. So the topography is generally a multi-scale topography. There's something at the micro scale and then at the nanoscale. And you can see that the contact angle for droplets is huge. So droplets will naturally roll right off. And sure enough, we can try to replicate, this is work from one of my PhD students, replicate some of those topographies in the lab. And I mean, you can think of this, Stellenbosch has great microfabrication facilities. Sure, there's no problem making complex pillared topographies. That's just really, science is there and it has been there for a while. The challenge is making those at scale, at sufficiently inexpensively and in a way that can be deployed in a pragmatic way that is durable and so forth. This is one of those examples that affects the microscale. I talked about something at the smaller scale, and these are, full disclaimer, these are just cartoons. There is an idea that at the sufficiently small scale, and then what I mean is that when the radius of curvature of those protrusions is commensurate with the thickness of the cellular membrane, that those might exert sufficient pressure locally onto that membrane to disrupt and spill effectively the guts out of the microbe onto the surface and effectively become biocidal, without using antibiotics. So only physical methods. You can ask the question, okay, well, if nature can do it, maybe we can do it too. Maybe it'll cost a little bit more, but over time we'll get there. So the first question to ask is, what topographies are those? What are those that are effective? And here's a relatively sort of complicated map that brings together. In fact, it's a simplified map because this one only brings together the periodicity, so something in microns, so the length scale of the plane, if you wish, and how tall they are, the amplitude. And the one that I'm missing here, but that's the third axis, is effectively the spacing between them. Because you can have very isolated spikes, for example, like in a nail bed. So it's not only the aspect ratio of the individual feature, the spacing between them, and how tall they are. You can see here, I've dotted some geckos and cicadas and dragonflies and things like this, just to see, what the challenges are. But typically, the idea here is that they need to be relatively high aspect ratio, tall structures, tall and slender structures, if you wish, that are more or less in the micron and submicron scales. So that's kind of the objective that I wanted to keep in mind, is that if we get that, we should be in business for at least, I'm not guaranteeing that they will last forever, but nature seems to do it just fine. This is on the left examples of existing sort of demonstrations on cicadas, dragonflies, and in fact, this idea of surfaces becoming biocidal seems to have some credence. This is disputed a lot in the literature, so I don't want to get into this, and that there are surfaces that can replicate that some effectiveness, but with the challenge of manufacturability. So let's ask that question first. Let's try to think of, let's say, a respiratory drop and see if we can, at the macro scale, see if we can get the surface to repel. It's extensively at scale, and I think we've already mentioned nanocellulose as the focus of this triple coat project. So we want to do those in materials that are sustainable. You've seen that just behind there we have diamond or black silicon or things like this. Those things are super expensive. They will never see the light of day. But nanocellulose seems to be a perfectly good candidate. And indeed, you can think of three types: nanocellulose, nanocrystals, nanofibrils, and bacterial nanocellulose, of which you can also make CNCs. And there's a bunch. It's not for you to pay too much attention, but it's just a complicated diagram to say there's lots of ways to fabricate on top of nanocellulose of the various types. So surely, if I combine the two ideas, I must be able to do something decent. That's the kind of the problem statement. One of the methods that we use is a method that kind of blows the surface up, it etches the surface with some kind of ionized gas. It doesn't matter which one it is. In this case, it was argon. You apply a voltage, these ions bombard the surface, and they will destroy the surface, but not arbitrarily. So, an example of what it does. Here you'll have the three types of cellulose that we consider. So from left to right, you have the time of exposure, if you wish. And you can see that at the beginning, the surfaces are, say, maybe quite planar. And then you have some kind of neat pattern that emerges. Those look very much like those biological patterns, I guess. And I think if you are maybe more like a physicist like myself, then you will do what's called a Fourier transform, which is what you see over here. And when you see a ring around, you know that there's a characteristic length scale that has been selected. So there's a well-defined periodicity. And then you might also see that the size of this ring is the company inverse the size of the topography. So you'll know that there will be some kind of what we call scaling laws. Things are well-defined. They have power laws. They are what we call fractal surfaces because they have a well-defined scaling, and that scaling is both in space and linguistic exposure. See whether we got there or not. We look at this with SEM, that's the images before. You cannot really quantify that well in 3 dimensions, but with, say, AFM or the cross-sectional techniques, you can. So if I look at this and I told you that I wanted tall structures, then I just need to do my maths and pick up something that has the aspect ratio that I require and the dimensions that I require. So works in that sense that at least we make the stuff that we wanted. And I guess the other thing I wanted to say is that the coating itself, even though length scale on the right side is 4 microns here, but the vertical dimension from bottom to top, let's see the numbers, but it's about a micron. So the coatings are exceptionally thin, such that they are actually transparent. So that's the other thing we wanted, was to be able to apply them on a window pane so that you can see through, right? Otherwise, it would not be acceptable to a consumer. Those are, this is my, I just had a big birthday recently, and so I decided to treat myself with a high-speed camera. That's really what I wanted to do. So this is it. I don't know if we needed one, but that's what I got. So this is what happens when you take the normal cellulose that hasn't undergone this treatment, and this is what you have on the right side with the treated cellulose. So the droplets do what we wanted them to do. They bounce right off. So this is about the dimension of a respiratory droplet. In this case, it's water, but we tried to load it up with proteins, with polymers, etc. They'll bounce right off, and once they stay here, because they need to end up somewhere, right? They could roll onto the floor or whatever, but they will stay with a spectacularly large contact angle. So they will be very easily removed. I told you that I wanted to impact the initial stage, so that was the first bit. But now, let's say that I end up with still a droplet on the surface, and something they need to go somewhere, right? And so the other bit and the other kind of length scale that we harnessed is one that is maybe more easily made through a process called wrinkling, which is not unlike our own skin wrinkling, which is what happens when you have an epidermis that is thin and stiffer, and a dermis underneath that is softer and more compliant. If these are irreversibly attached, then as things get deformed, something needs to give, and mathematical solution to this is that they will undulate. I'll show you in a second why that's such a natural consequence. But this is just to tell you that actually man-made, so these are all man-made by us, and you can make these beautiful things spontaneously over very large scales. So can these topographies, now you can see these numbers, the length scales now are here in maybe few microns or maybe tens of microns. They are now commensurate with microbes, for example, and you make them commensurate also with fungi and so on. So why is it so natural? Just take a soft material, you place the epidermis on top, the thin, stiff skin, you apply some kind of deformation, and they should undulate if they are adhered properly. I mean, if they're not, they will simply delaminate, which is what happens normally, right? But that's why what Alexandra was presenting is important. If you make them stick to each other, they will not. And then what they will do is that the compliant one is happy to accept the deformation, but the rigid one is just not happy at all. But because they are married together, they will negotiate some undulation on the surface, and that, as a mathematical problem, gives you a selection of a wavelength. The way to make those, I mean, in the lab is easy. For example, if you can take a ubiquitous material like siloxane and you generate the glassy skin on top, you have a huge stiff on soft kind of what we call a bilayer. And then this process is very well behaved, so we can program it nicely. The thickness of that skin can be designed very nicely. And then the mats actually are back from the 18th century. So they're really, here the novelty is that we are bringing these into the nanoscale, and we are deploying those to address a kind of societal challenge. So this lambda is the wavelength of the surface, so the periodicity between peak to peak. And what you'll see here is that there's H, which is the film thickness, which is why I was saying it's important to control that skin. The thickness of the skin largely you see has an order 1 with the wavelength, so they're directly proportional. So if you design the H, the thickness, you pretty much have the lambda control. So then the amplitude over here responds to this, everything else the constant except for this epsilon, which is the strain, how much you compress. And intuitively, the more you compress, the taller they get. There's no more to this really. And it really works, and it's such a robust process. Of course, the mats can get hairy and there are perturbations, etc., but the basics is this: the periodicity comes from the thickness, and how tall it is comes from how much you squish it. And there are other ways to do this, by the way. You don't have to do it just mechanically. For example, you can do it simply by applying a coating. And the drying process will create a shrinkage that makes it work. So you can apply temperature fields. There's so many ways to do this, but we often like to use mechanics because with mechanics you can do this trick lots of times. So that's what one of my students did. You see here, you can create first the generation that just gives you a sine wave in one direction. And then you apply a second wave, and you can get a second generation that creates this kind of spiky surfaces. That's kind of what we wanted to copy in our lecture. So I don't know if those of you who like marshmallows, you will remember or maybe music as well. There's an analogy here with music with the Fourier transform, which is frequency analysis of a sound. If you take a sound wave, it's a pure sound wave, it has only one frequency. And here we have one frequency in one direction, the other frequency in the other direction. And you've created in this way like a spiky surface. So that's exactly this. This is a two-dimensional surface that you can design to become spiky. It's like think of it as an egg box, perhaps, or a square array of two waves that have been superposed. And this process really works very nicely. So I told you that I wanted the droplets, if they were to stay there, to roll off, to get out of the way. Right? We spoke about the droplets that should not attach in the first place, but if a droplet were to land there, the question that Hisei, also in our triplet team, has asked is, what do I need to get a droplet to just fall if it happened to land there? And so here we can come back to the same suspects. Amplitude was the one we were trying to design earlier. And the thing is a contact angle. So this is some intrinsic property of the material itself. So he says put here together that says, okay, my material might be nanocellulose, might be steel, might be glass, might be polyurethane. These are the materials that we were preoccupied, for example, with what we call these high traffic areas, like in a bus. You could think of holding the handrails, and those would be maybe some kind of metallic surface, maybe steel. You could think of the glass. You could think of maybe the fancy plastics in cars that are often polyurethanes. You can think of the fabric on the seat that could be maybe a polyester thread, perhaps. And so that's what happens on this angle, on this axis. So those are the intrinsic properties of the surface. But on the horizontal axis is just the height of the material, of that pattern. And what you can see here is that in this region, if you make these guys sufficiently high, if the contact angle is sufficiently low, then these guys will simply roll right off. So it's again it's one of the kind of first points of intervention, that you can make these things not only bounce, but also if they were to land there, we have now a mathematical tool to tell us what's needed to get potentially infected drops to slide off. This depends on the pattern itself. So this has looked at the orientation of the pattern. For example, if you make the life easier for the droplet to roll, as in if the grooves are oriented south, they will roll. But if you make it hard, they might actually stick, and everything else being kept the same. And it turns out that he was able to kind of generalize all of this. So maybe there's more to this map than I have time to explain you. But let me tell you something that I think you'll find exciting, which is that drops have shapes that are normally hemispherical cups, but those depend very much on the topography itself. So in this case, I have something that's flat, and therefore the droplet is, the top view is a circle. But you can see that they can get quite funky very quickly, depending on what pattern you're running. And he says in writing, this is why I love this type of work that we do. This is so interdisciplinary. He's asked the question about, can we really correlate the pattern where they are sitting with the shape of the droplets and their roll-off? So it's really the first time that I think really anyone has tried to tackle this in a truly general way. So there's maths involved here, because he calculates now the effective interfacial energy functional, and he does some kind of a path integral along the contour of the droplet. So leave this here just for us to have a chat maybe over coffee later if that's of interest to any of you. But the process is very simple. It says I can take a droplet that is initially circular, and I can apply this mathematical tool to say you have perturbations on the shape of the droplet. So a droplet that started in a circle and then it starts becoming elongated until, for example, this one becomes rectangle. This one has lots of, this looks like the Citroen sign, I think. I mean, you're trying to try a Citroen, don't you? And so the Citroen is the chevron pattern, and it, in fact, the contour of the droplet is decorated by all these Citroen signs. And so it turns out that you can quantify these perfectly. And again, those things not only affect the shape of the droplets, but if we make those dimensions commensurate with Imperial or even size of fungi, they will become effective. So this is something that has been, we've been scratching our heads with the team at 311 since started about reproducibility of results and surfaces, these intrinsic and what we call extrinsic properties. But here's just an assortment of data from the previous publication where we looked at the effect of proliferation of some model by model microbes here. The key result that we found, the key result that we found here was that effectively these surfaces, the ones that are what we call checkerboards, these chevron patterns, were the most potent. And that we found that the local tortuosity, the fact that they had sharp angles that were commensurate with the microbe sizes, quantifiably decreased their proliferation rate. But maybe in a more qualitative way, we felt could impede the colony formation because they will disrupt the local packing order by preventing those to order, for example, in a regular array, like we would otherwise, not topographically patterned, in ones that are patterned, but in ways that actually allow them to connect in a very energy low cost way. So can these be done into nanocellulose, and how can we make these at scale? The answer is yes, too, and this is the work that Hui has been doing. This is just an idea that once you've made a print, if you wish, you can copy that onto any material. You can just use it as a stamp. It's perhaps easier said than done, but this is a cartoon of how Hui does it. She makes first the mold, then she takes a nanocellulose solution, and then the trick here is that she stamped it onto a support that allows both the transfer to be made and the film to form. So it dries as well as stamps at the same time. And this is really a breakthrough because people have not been able to do this. And then she's also demonstrated, once you've demonstrated the first step, then you also know that you can roll these across. So you can make those at scale for sure. Once you have the first, think of a pasta machine or a roller in the kitchen, you can make those. If you make it once, then you can continue making those if you have the appropriate surface properties. So this is what was in her most recent paper, just to say that actually the nanocellulose, especially these chiral nanocellulose, can make really interesting structures. But here she's able to make these within about 10 minutes, and a fidelity with a pattern of in excess of 90%. So it's working very well. And this is a demonstration of just the various patterns that actually have to be transferred onto nanocellulose. So that also seems to work well. And they seem to survive pretty well. So this one of the concerns that we had is that if we have it on a surface that is you sneeze over or it's raining over this, will it last? So this is a diffraction experiment where she's taken pattern, she's placed some simulated raindrop on top with some salts, etc. And then you'll see that over time you recover the diffraction pattern that you expect for a regular period, like a regular pattern like this. She's taken this to hundreds of humidity cycles, and the pattern sort of responds to humidity because cellulose is hygroscopic, but always seems to bounce back and recover. And over many months, over like a whole year, they have stayed. So they're robust. Now, we've been working with the team at K11, Gina, Hans, etc., trying to find out what exactly is the activity of the patterns. And the data, the jury is still out, so we don't know what's going on there. So I'll skip this one for now. Let me tell you only perhaps to wrap up that nature, of course, I'm not an evolutionary biologist by not a stretch of imagination, but these patterns, they have multiple functions. So I focused here on a function that was this antimicrobial function, but there are a whole host of other people, including also in my own group, that look at these very same patterns for different purposes. So, for example, you will all be familiar with this, the structural color of some of these very same surfaces of insects and plants, etc. This is an example of a morpho butterfly that uses exactly the same patterns. The wavelength is commensurate with the wavelength of light, which is exactly the wavelengths that we're interested in. And this is with the exact same material, the result of what you can achieve. So you dial the color to become. I told you that you can dial wavelength and amplitude. You dial those two numbers to get the wavelength that you want. The surface can be antimicrobial and potentially really beautifully colored for whatever purpose. For people interested in paints, for example, or of course care about both of these aspects. This is just how mathematical the whole thing is. This is high school physics, if you wish. This is the periodicity of the structure. This is theory, this is data. Periodicity of the structure, the angle of observation, you use Bragg's law to calculate the angle of diffraction, and boom, you get the color that you should rightly expect. So, in a sense, this is what I guess gets me with these methods, is that the science is there and is established, but the engineering, so the development of this at scale, sufficiently low cost, in a way that is deployable, durable, etc., is where I think the innovation steps are. I'll stop here and just acknowledge Hui and Hisei, who've done the work. Hui concentrated mainly on the cellulose pattern, and Hisei on the science of the wetting and spreading. We've been really enjoying this work with the Triple Consortium, and especially on the antimicrobial test, work closely with Dina, Sam, Hans, and Ben at Imperial, that benefited from samples, advice, etc. from everyone. I'll leave you just with maybe the three thoughts. The first is that this nanoscale topography, nano and microscale topography, is incredibly effective at tuning wetting and spreading. It's not new, but it's something that might be relevant for us in this community of antimicrobials, because this is the initial contact. Will a drop stick to that or not? Can you get to do something that will make all of the subsequent steps faster, easier, better? They will eventually, microbes win over for sure, but can we maybe make the first step easier, and that initial intervention is effective? The second one is that if a droplet were to sit there, say a respiratory droplet, for example, can we make them go away? Can we get them to slide? Can we get them to do things that they would not otherwise do? And the answer is also really convincing yes to this one. The final is, we need to make the materials sustainable for all sorts of reasons, and nanocellulose is a perfectly good candidate for this. And you'll hear in the other talks many modifications. Here we've used neat nanocellulose, but all of these can be augmented by the chemical strategies, chemical and biochemical strategies that here we've not covered at all. So this is just, the neat. So we often produce our own from biomass waste material. So there's no chemical modification to this. And even in these cases, we can show that the rough material can be patterned at those nano to micron scales. And so the next steps for us is to augment these strategies from the physical to the biochemical to get sort of increased potencies, etc. Stop over here.